next speaker is Dr. Gül Dölen. Dr. Dölen earned her MD and PhD at Brown University and Massachusetts Institute of Technology, MIT, where she carried out seminal work on the pathogenesis of autism. Dr. Dölen completed postdoctoral training in the Department of Psychiatry at Stanford University, where she did paradigm-shifting work on the neural circuits underlying social reward learning. In 2014, Dr. Dölen began her faculty position at the Department of Neuroscience at Johns Hopkins University School of Medicine. And her laboratory studies neurotransmitters, brain circuits, developmental programs, and the evolution of social behaviors, with a focus on diseases of social brain, including things like autism, schizophrenia, PTSD, and addiction. And this is where the connection to our theme of psychedelics today comes in. Recently, her lab has become interested in uncovering how MDMA induces pro-social behaviors across species, including humans, mice, and octopuses, as well as shedding light on the mechanisms underlying MDMA's profound neuro and therapeutic effects. Dr. Dölen is the recipient of several prestigious awards. She is widely cited and very well respected in our field. It is a great pleasure to introduce her to you today.
Thank you. All right. Thank you very much. That was so nice. It's wonderful to be here. Let me just make sure I got this clicker thing. So hi. I just tagging on from the last talk. I did actually give a talk at NASA a couple years ago and tried to convince them to let me give MDMA to astronauts. They said no, but you know, it's a work in progress. I might go back in a couple years. Okay, so today I'm going to tell you a little bit about some work that's already been published, and then I'm going to tell you about some new work that is unpublished that I really think is particularly exciting for this group. So as was mentioned, my lab works on social behaviors from a variety of different perspectives. I'm not going to stand over here so I don't have to keep looking up. And I'm not going to talk about the octopus research at all today unless you ask me during the question answer. Instead, today I'm going to focus on our work on development and critical periods. My talk is a little bit shorter than I had prepared for, so I'm going to skip through some things as we go through, but I'll just introduce the idea of critical periods here because I know that not everybody here is a neuroscientist. So neuroscience has been obsessed with the idea of critical or sensitive periods for almost 100 years. They were first described by Konrad Lorenz to describe imprinting behavior in snow geese, but there are a lot of different critical periods that we know about. So song learning in birds, mapping of the somatosensory cortex. The somatosensory cortex received the Nobel Prize this year, so exciting and cool work going on there. Also human language, so anybody who's tried to learn a language later on in life knows how difficult that is and that you always learn with an accent. This is because there's a critical period for language learning. Probably we know the most about critical periods, mechanisms from pioneering work by Hubel and Wiesel and many, many others that won the Nobel Prize for ocular dominance plasticity, which is a type of learning and memory that happens in the visual cortex. And then clinically relevant critical periods for things like motor learning after stroke, okay? So there are these windows of times right after stroke where you have your maximum ability to learn, and after that window is closed, you're done. And so you're not going to recover much more function after that window is closed. So these are really, really important, not only for understanding how the brain works, but also neuroscientists have had the intuition that, maybe we keep figuring out all of these mechanisms for how different diseases work, but then when we go to clinical trials, they just fail. And maybe the reason that they keep failing is because by the time we intervene, the relevant critical period has already closed. And so neuroscientists have been really interested in trying to figure out ways to reopen critical periods. I'm just going to quickly point out that this is a sort of diagram illustrating the critical period for ocular dominance plasticity. I'm not going to go through the details here. I just want to point out that as the brain matures, as the animals mature, the learning and memory that's possible, or the neural circuit arrangements that are possible in the brain, peak kind of around usually around juvenile, adolescent period, and then decline over time, and then they're closed. And people have identified mechanisms, brain mechanisms, synaptic mechanisms, plasticity mechanisms, circuit mechanisms, both the decrease of which and the increase of other factors that seem to constrain this learning and memory to a specific window of time that we call the critical period. And as I mentioned, neuroscientists have really been focused on trying to understand these mechanisms, partially so that we can figure out methods for reopening them for therapeutic benefit. Okay, and people have identified a number of different manipulations that do this. And to be honest, when I first started working in critical periods when I was a graduate student, you know, I was very, very skeptical of the idea that there might be a sort of universal master key for unlocking critical periods in the brain. In fact, I remember saying at some point that even if we ever found something like that, it would just melt the brain and cause seizures and be a terrible idea, and that we would never want to do that. So my lab, when I started my lab, we discovered a new critical period. So this is the critical period for social reward learning. We measured in mice. This is just a plot, a regression plot, fitting the data from 15 different ages. This is just the male mice, but we have the same, almost the same exact plot for female mice. And I'm not going to go in through the details of how we measure this social reward learning, but just to give you a sense of what this might be like in humans, social reward learning is, you know, the reason why teenagers are so much more susceptible to peer pressure than we are. When they're young, they're learning their world, they're learning from their peers, they care a lot about the social dynamics within their peer group. And it's also why when we get older and we travel to different countries, we feel a little uncomfortable. You know, whenever I go to Japan, I feel like I'm a bear in a china closet. And that's because I learned the social rules for this culture, and I'm a little bit less familiar with the cultural rules in other cultures, okay? So this critical period for social reward learning, we think, is interesting for a number of different reasons, and I'm happy to discuss this further, but in the interest of time, I'm just going to skip ahead and show that we just discussed that we related this behavioral learning and memory critical period to a form of synaptic plasticity in the nucleus accumbens, which is a part of the mesocortical limbic reward circuit of the brain. We did these experiments using whole cell patch clamp electrophysiology. And basically what we're doing is we're measuring a form of synaptic plasticity that is induced by the peptide hormone oxytocin. And what we showed is that when we give this manipulation in a acute brain slice, so a slice of brain that we've cut and kept alive using artificial cerebrospinal fluid and oxygenation, we can induce this plasticity by bath applying oxytocin, and that ability to induce oxytocin-mediated plasticity in the juvenile mice is very robust, but when we try and induce the same plasticity in adulthood, we are unable to do so. And so that change over time, over development, in the ability to induce plasticity is what we call metaplasticity. And that's a really important term. I know people in this audience have probably heard the term plasticity, but I want to start to get you to think a little bit more in a nuanced way about plasticity. So this is metaplasticity, so the ability to induce plasticity, not just plasticity writ large. This was specific to the oxytocinergic type of plasticity in the nucleus accumbens, so serotonin plasticity is not developmentally regulated in this way, even though serotonin is also required. And we thought we would like to be able to reopen this critical period, and we thought, well, given all of the sort of recreational and sort of underground knowledge about MDMA's ability to have these incredibly profound and very, very robust pro-social acute subjective effects of MDMA, that maybe this would be a good way of trying to reopen this critical period. And I'm not going to go into any of the other scientific reasons that we had for choosing this, but we started with this. And what we showed is that if we gave MDMA and then waited for 48 hours. So we're not measuring the acute prosocial effects of MDMA the way we did in an octopus or that other people have done in rodents. We're waiting. So we give it, and then we wait for the acute subjective effects to wear off, and then 48 hours later, we measure social reward learning in these mice. And what we find is that in the animals that were pretreated with MDMA, but not the animals that were pretreated with saline, we get a return of the magnitude of social reward learning in adulthood that we see in juveniles. And for a variety of other control experiments that I'm not showing you, we think that this is evidence that MDMA reopens this critical period for social reward learning in mice. Okay? We were also able to relate this back to those metaplastic changes I talked about with oxytocin LTD, because when we did this pre-treatment with MDMA and then cut slices 48 hours later and then measure oxytocin LTD, then we get a return of this juvenile level of oxytocin-mediated synaptic plasticity. So MDMA also induces a metaplastic upregulation of oxytocin plasticity. I want to differentiate metaplasticity from hyperplasticity. And this is really important, especially for those of you who are interested in some of the medicinal chemists' approaches to trying to find new ways and new drugs. We need to be careful about this because hyperplasticity is something that cocaine induces, right? So we know that cocaine has this robust ability to induce kind of not synapse-specific, robust general plasticity all over the brain. And we think that that hyperplastic profile of cocaine, which we see in the nucleus accumbens, the medial prefrontal cortex, all over actually the brain, is related to this ability to induce this non-specific, generalized hyperplasticity. And so it was really important control for us to know whether or not cocaine is also able to reopen this social reward critical period, and it does not. And so to us, this is a really important clue that because cocaine doesn't reopen the social reward learning critical period, that this is one of the things that is different between cocaine and psychedelics is also their therapeutic potential, right? So cocaine does not have the same therapeutic potential as psychedelics for treating diseases, neuropsychiatric diseases. And so this was one piece of the evidence that this critical period mechanism that we're describing underlies the therapeutic effects of these drugs. There was some other evidence as well. So just like the therapeutic effects that people like the Mithoffers have described, the therapeutic effects of MDMA are rapid, and they have these very durable, long-lasting effects that go well beyond the acute subjective effects of the drugs. We also were able to show that MDMA in a mouse reopens the critical period. It starts to happen as soon as we can start measuring it. So while they're having the acute trip, it's very difficult to measure this. But at the 6-hour time point, we already see it coming up. By 24 hours, it's significantly up. And it lasts for 2 weeks after we give the MDMA. And then by 3 weeks and 4 weeks, it comes back down to baseline. Okay? So that's interesting. And then we already heard about this, and I think this is really, really something that differentiates our model, our neurobiological explanation, from all the other explanations out there, right? So the therapeutic effects we already heard are context dependent, right? So people in this room are excited about psychedelics not because they are next generation anxiolytics or next generation antidepressants. These drugs might have those properties, but the really profound sort of one-and-done cure types of things that we're all so excited about relate to this ability of the drugs to only work in the right context, right? After all, people aren't taking MDMA, going to raves, and poof, their PTSD is cured. They actually have to be in the right set and setting. We can't measure set in a mouse, but we can change the setting. And so what we found is that the ability to reopen the social reward learning critical period with MDMA is also dependent on the social setting. And so to our knowledge, this is the first time anybody's been able to recapitulate this property in an animal model. Okay, so MDMA is just one of many psychedelics that we're all here to hear about. and the medicinal properties of these psychedelics in an increasing number of clinical trials. I just listed 4 here for things like depression, addiction, PTSD, addiction again for psychedelics ranging from psilocybin, MDMA, ibogaine, and ketamine. Okay, and we already heard they have medicinal properties that we've known about for millennia, and despite the fact that we've known about this for a long time, we really don't know what is the mechanism underlying it, and I would propose that this reopening of the critical period might be an important clue. And so the reason that we were a little bit skeptical of this when we first started these experiments about 4 or 5 years ago is that, MDMA is a little bit different. Even though all psychedelics share this ability to change or alter the state of consciousness, MDMA is unique in having these very robust prosocial properties that, the other psychedelics don't really necessarily share. People who are doing psilocybin or ketamine aren't going into cuddle puddles. They may, after, a couple of days, come back and say they feel connected to the universe, but the acute subjective effects really are very, different across psychedelics. And so if it was the acute prosocial effects of MDMA that was responsible for our ability to reopen the social critical period, then our prediction would have been that ketamine, PCP, LSD, psilocybin, ibogaine would not reopen the critical period. But if we were wrong, if it was that MDMA's ability to reopen the critical period had nothing to do with the empathogenic qualities of acute prosocial qualities of MDMA, but was instead sort of what it feels like to reopen a critical period is the same as what it feels like to enter an altered state of consciousness, then our prediction would be that all of the psychedelics would do it. And so we started by just looking at all the psychedelics. So we started with psilocybin. We did the exact same experiment with MDMA. So we give this psilocybin, we wait 48 hours, and then we measure social reward learning. And just like MDMA, psilocybin reopens the critical period. LSD, same thing, reopens the critical period. Ketamine and ibogaine also reopen the social reward learning critical period. So this is exciting, but we wanted a little bit more evidence around this. So one of the things that people have noticed about psychedelics, this is from the human literature, I'm just cartoon diagramming roughly based on these various publications, that the psychedelics vary dramatically in the duration of the acute subjective effects, right? So ketamine is very short-acting, 30 minutes to 2 hours. Psilocybin and MDMA's effects, acute prosocial effects, last anywhere from 3 to 6 hours. LSD is a little bit longer, 8 to 10 hours. Ibogaine is sort of the rock star of the group, 36 to 72 hours, really, really long trips. And the time course of these acute subjective effects, there is some evidence is also proportional to the durability of the therapeutic effects, right? So ketamine is rapid, but it doesn't last. So typically the antidepressive effects of ketamine wear off within a week. Whereas MDMA, psilocybin, LSD, ibogaine, these are having long, durable effects that might be related to this time course of the acute subjective effects. So we wanted to look at the time course of the ability of these psychedelics to reopen the critical period. So we gave the psychedelic drug, and then we measured 1 week, 2 weeks, 3 weeks, and 4 weeks after the psychedelic, and just mapped out this time course. And so what we found is that ketamine, just like even though it reopens at 48 hours, at 1 week post-injection, it's no longer opening the critical period, but psilocybin does. At 2 weeks, LSD and psilocybin both keep the critical period reopened. At 3 weeks, LSD but not psilocybin reopens the critical period. And at 4 weeks, ibogaine but not LSD reopens the critical period. And so what's remarkable is that when we plot these results on this figure, again, we see this proportional response. So ketamine reopens at 48, closed at 1 week. Psilocybin and MDMA reopen at 2 weeks, closed at 3. LSD open at 3, closed at 4. And ibogaine, we've only ever measured out to 4 weeks, but keeps it open for a month after the critical period, after we give the drug. And this result has a number of implications. First of all, I think it adds to our evidence that this is what it feels like to be this altered state of consciousness is related to what it feels like to reopen a critical period idea. Second, I think it tells us that we want to be careful about giving these drugs and then just releasing people out into the world. I think the insight that, we need to have this reintegration period from the clinicians is relevant here. But also I think that we need to be very careful about what kind of setting we introduce people into, especially if these drugs are having these long-lasting effects. And that the third thing is that these long-lasting effects might have something to do with their therapeutic efficacy. And so any efforts that are being, pursued to limit the duration of these acute subjective effects could interfere with the therapeutic efficacy of the drugs. And so you could be taking a drug like ibogaine that is incredible and being able to help resolve, really profound heroin addictions and turn it into something like ketamine, right? So we want to be careful, and the more we understand about the mechanisms of these types of long-lasting effects, I think the better. So how much time do I have? 5 minutes. All right. Okay. I'm going to go really fast for the rest of it because it's a lot of mechanism, and I don't want you to get overwhelmed by it. I'll just point out that LSD also induces a metaplastic upregulation of oxytocin, just like LTD. I also want to kind of not debunk, but just sort of raise some questions about universal mechanisms. So we're really interested in universal mechanisms, and I know a lot of people in this room are heavily invested in the idea that everything comes down to serotonin 2A. Sorry, I see Manoj. We had an argument about this yesterday. And I'll admit, when I started in this field, I saw the serotonin molecule and the LSD molecule right next to each other. I was an undergrad, and I was like, aha, this is how we're going to understand consciousness, right? Like, I was so excited. And so the serotonin hypothesis is something that I have, been excited by for a really long time. There's some evidence that that's binding to the serotonin 2A receptor by LSD triggers beta-arrestin signaling, which I don't really need to get into the details of that. But the psychedelics as a class don't just target the serotonin 2A receptor. So there's lots of evidence that there are other receptors that are important for the therapeutic effects of these drugs and for the psychedelic effects of these drugs. So we just wanted to look. And so what we did is we used the same serotonin 2A antagonist that people have used in humans, ketanserin, and just asked whether or not it could block the psychedelic's ability to reopen critical periods. And so, yes, the serotonin 2A receptor is required for LSD's ability to reopen the critical period. Same with psilocybin. Psilocybin requires the 2A to be able to reopen. But neither MDMA nor ketamine require the 2A receptor in order to reopen this critical period. So it's not the universal mechanism that everybody has been hoping for. Similarly, beta-arrestin 2 knockout mice. So beta-arrestin 2 is a second messenger signaling cascade. There are mice where it's been knocked out. We did control experiments to show that those mice are basically normal. But when we looked at LSD's ability to reopen the critical period in these knockout mice, they were not. So beta-arrestin 2 does seem to be required for LSD's ability to reopen the critical period. Interestingly, MDMA also, but not ketamine and not ibogaine. Okay, so beta-arrestin is not the universal mechanism that we were hoping for. So I know this is a really complicated slide. Don't get scared. The idea is to just let you know that it's complicated, and it's not going to come down to a single molecule or receptor or second messenger signaling cascade the way that we had hoped. Instead, what we proposed is that the mechanism that's universal across all of these psychedelics must be downstream of the receptors, downstream of the second messenger signaling cascades, and we proposed that they must be, it must be happening at the level of transcriptional regulation, DNA transcription, regulation of DNA transcription. And the reason that we thought that is also because these effects are very, very long lasting. So I know this is a big slide. Again, don't get overwhelmed. What I'm trying to get across here is that when we took advantage of the fact that we knew that cocaine does not reopen the critical period, and that different psychedelics keep the critical period open for different amounts of times, then we can do this transcriptional profile and compare genes that are changed in their regulation, so upregulated or downregulated, in the open state versus the closed state. And when we do that, we came up with 65 genes that are differentially expressed in the nucleus accumbens under these conditions, and roughly 20% of them were regulating either immediate early genes or extracellular matrix proteins. Okay? And if we didn't do that control, if we just did drug versus cocaine, for example, we didn't come up with any significant gene ontologies. We just came up with like random, the synapse is involved, the brain is involved kind of genes. Okay? So it was really important to be able to define the critical period open state versus closed state in order to come up with significant gene ontologies. And the reason that I'm so excited about these particular ontologies, this extracellular matrix and the immediate early genes, is because if you remember from my introduction, when people were trying to understand how critical periods get closed, some of the things that have kind of been proposed as universal mechanisms and things that people have tried to target for therapeutic benefit were extracellular matrix proteins and immediate early genes, even though those were identified for cortical critical periods, and this is an accumbens critical period, right? So it seems to be sort of going across different types of critical periods, so sensory versus emotional. And interestingly, there is a report, and these people who did this study were actually, I think, mostly concerned about side effects of ketamine and like important controls you need to run. And what they showed is if you give 6 back-to-back doses of ketamine, you can accidentally open the critical period for ocular dominance plasticity. And so what I think we might have actually discovered is that psychedelics might be that master key for reopening critical periods that that I never believed in until I saw these results. And that the specialization here might be that it's not melting the brain and causing seizures because it has this other requirement of the set and setting dependence, or for a neuroscientist, we call that experience dependence or activity dependence, and that having this co-requirement of only being able to change the synapses in a setting-dependent or context-dependent manner is why psychedelics are so exciting. So I'm just going to skip ahead. I'm not going to show you, some of the studies that we're pursuing right now to try and use this for other diseases like stroke. I'll just give you the sort of plug, and you can look this up on our website. This broad class of being able to extend this idea of reopening critical periods outside of just neuropsychiatric disease. Sorry. I'm not sure why this is advancing. We're calling it the Fathom Trials or the Fathom Project, and it's psychedelic healing, adjunct therapy, harnessing open malleability. And we are pursuing this in a number of different avenues that I think really expand the scope of diseases that we can think about using psychedelics beyond just neuropsychiatric disease and really start going into neurological diseases as well. And so with that, I just need to thank the people who did this work. Roman Nardu is the extremely talented postdoc in my lab who, really was the lead on all of these projects. A number of other people in the lab have helped out with this project. Genevieve Stein O'Brien is an amazing mathematician and computational neuroscientist who did the regression analysis and also the transcriptional profiling. She's a postdoc in Loyal Goss Lab. And then I have to thank all the people who gave us reagents. And I didn't talk about the optogenetics work related to this, but Ed Boyden's lab, who made us some cool tools. And then, of course, funding support. So with that, I'm done.